Here we go. Okay, so what we have are two diagrams side by side, one that goes through glycolysis and one that goes through the Krebs cycle or citric acid cycle. And it's basically looking at metabolism from the point that glucose enters the system. I made a smoothie for my kids at lunch today, and they weren't drinking a glucose smoothie. They were, you know, drinking fruit and yogurt, and now, you know, after there's some chemical breakdown in the stomach, at some point, some of those molecules are going to be glucose molecules, and some of them would be, you know, lipids and proteins that get used to make biological macromolecules for cells and tissues, and some are going to go directly into making ATP. And so for the purpose of Biology 160, and looking at aerobic respiration and looking at glycolysis, we step in at the point where the body of a eukaryote has made the molecules it's ingested into glucose. And so we just kind of fast forward to that point. And if you remember from chapter 2, glucose is a 6-carbon molecule that tends to be circular when it's in fluid. So in cells, that's how you find it, not as a chain, but as a circle, as a ring. And what happens is, in order for the body or for cells to capture the large amount of energy that's between each of those carbons in those covalent bonds, it has to modify the glucose step by step. And as it modifies the glucose, it first uses ATP and then liberates it or produces it. So let's see. This is where I get to have some fun with scribbling again, and I want to try to make sure that it stays on. So right here, you see the ATP is used, and then I have to do it again here. Right here, you see that ATP is used. Oh, that's hilarious. Okay, let's try that again. I must have clicked something. Okay, so we go from glucose to this glucose 6-phosphate and then to fructose 6-phosphate. And if you can see here on the other side of my scribble, it's still a 6-carbon molecule, right? But it's been phosphorylated, right? And in fact, after we use these two ATPs and they become ADP, the 6-carbon molecule has 2 phosphates attached to it. And what that does is it makes it very biochemically unstable. Phosphate with the phosphorus in the middle and the 4 oxygens attached to it is really unstable, really chemically reactive. And when you phosphorylate a molecule, you make that molecule chemically reactive or unstable, and thus able to participate in a chemical reaction. So glucose by itself, it has a lot of energy stored, but it doesn't just break down and, you know, burst into flame on its own, even though breaking it down would be an exergonic reaction that would release energy. And so in order to get glucose to break down at body temperature, we have to destabilize it. And we do that by consuming some ATP, investing it into the system. And so now we're down to ATP at a cellular level, but we've destabilized the glucose, and we've converted it into something else. And then do you see the 2 NAD making 2 NADH right here? That NADH
Can I ask a quick question? Sorry. So when you do the glucose and fructose and you use 2 ATPs, how come you only have 1 phosphate attached then?
It's being called carbon 6-phosphate or fructose 6-phosphate right here, but you don't actually, it's not showing you structurally the whole thing and what it looks like. We phosphorylated it twice, but we don't call it diphosphate. It's just fructose 6-phosphate. And it has to do with now there's 2 phosphates on there.
Okay.
Good question.
Thank you.
So the next thing that happens is because it's somewhat unstable is, more phosphate, but not ATP, phosphate available in the cell gets, we basically put in protons, we put in some energy, and by doing that, those protons are hydrogen ions. So we're not using ATP, but we're using hydrogen ions available in the cytoplasm, and we turn NAD plus into a product, NADH. And by bonding this, and it's nicotinamide, I can't even remember how to say it right now. We'll get there in the PowerPoint. That's sort of embarrassing. You don't need to know how to say it, but it's a long name that basically means this is a temporary energy carrier. And so when the hydrogen covalently bonds with the NAD, the covalent bond is storing a little bit of energy, and this NADH gets sent into the mitochondria as an electron transport carrier. It carries electrons and it carries hydrogen ions, and it drops them off later to be used to make a chemical and electronic gradient. And so instead of just burning glucose up all at once, we use a little bit of energy, and now we're starting to capture energy. And if I circle again here, you see now we make 2 ATP. So minus an ATP makes some NADH, and now we make 2 ATP. And do you see over here the 2X and the 2X? What that actually means is at the third change, the third chemical reaction that happens, actually splits the original glucose into 2 3-carbon molecules. It's been split in half. And each of them allows us to create 1 NADH molecule. And so between the 2, we have 2 NADH. Then we take those 2 3-carbon molecules that originated from the glucose up here, and each of those goes through a chemical reaction that produces an ATP. And so we get 2 ATP. And so now minus 2 plus 2, we're at 0, right? We've harvested 2, but we've used 2. But what happens in the next couple of steps? It changes again, and it releases water, and it changes again, and it releases ATP. Each of the 3-carbon molecules releases another ATP. And so what we find at the end of glycolysis is that we've invested 2 ATP, and we've harvested 4 ATP, and so our net gain is 2. And the reason that that's important is glucose by itself, when we break it down through glycolysis, making 2 ATP isn't a whole heck of a lot. It's a small fraction, actually, of the total energy stored in a glucose molecule. But we're not done. Do you see this gray arrow down at the bottom? And how the molecule that is now pyruvate, and there's 2 of them from each glucose molecule, that pyruvate gets pulled into the mitochondria. And the pyruvate then gets broken down further in the citric acid cycle or Krebs cycle. And then the energy carrying molecules that came from glycolysis, the NADH, and these NADHs and FADH2s that came from the Krebs cycle, those get pulled into what we call the electron transport chain in the inner membrane of the mitochondria. And as they drop off their hydrogen ions and their electrons, they create a biochemical gradient across this inner membrane of the mitochondria. And then that concentration gradient is used to make approximately 34 ATPs, all from the original glucose molecule. So how many ATPs do we get just from glycolysis? 2. How many more do we get when we finish breaking down the products of that glucose molecule in the mitochondria? Another 34 to 36, actually. So it's 17 or 18 times more energy obtained from just one glucose molecule if we finish the breakdown by using the mitochondria. So see how mitochondria help use energy efficiently in a eukaryotic cell? Do I still have you all with me?
Yeah, that makes sense.
Yeah.
Okay. When I can't see your faces, it's hard. Okay, so basically chapter 4.2 through 4.4 walks you through glycolysis and then aerobic respiration. And the reason we call it aerobic is because this whole last piece right here in the electron transport chain, I'm going to circle it right here. Do you see the one? Oh, that's hilarious. I did it again. Okay, let's try one more time. Nope, not gonna work. Okay, so maybe if I just try to underline it. That was really bad. It's 2H plus 1/2 of an O2, and then this arrow to the right yielding H2O. What that is showing you is that every time we send hydrogen ions through the proteins embedded in the mitochondrial membrane, and we make water, which is why you sweat when you're burning energy. The only way to make water and the only way to pull these hydrogen ions through and to capture the energy to make ATP is if we have oxygen ready to combine with electrons and hydrogens to make water. If oxygen is not there, this whole process of transferring electrons and moving hydrogen ions across, it comes to a screeching halt. And so if oxygen is not present, then the electron transport chain doesn't run. And so then we have no gradient and we can't make ATP. And if ATP isn't being made in the mitochondria and this backs up, then you can picture this backing up too, the citric acid cycle. And so then what are you left with if there's no oxygen as your only means to making ATP for the cell to use to make proteins work and perform cellular work? Then the only thing that you're left with is glycolysis in the cytoplasm. And it will continue to make ATP, but 2 ATP compared to 34 to 36 ATP is a really minuscule amount. And so you can't function very efficiently on glycolysis alone. And in fact, without aerobic respiration, we end up with another problem where we end up with this inability to continue with glycolysis past this point if we can't make NADH. And the way that, it's really important for the cell to finish glycolysis is to have NAD plus available. And if the cell runs out of NAD plus, then it can't finish glycolysis. And so that's where some cells have fermentation, a process that burns through NADH to make NAD plus when aerobic respiration isn't happening, when this right side is effectively crossed out. And so I wanted to show you these two side by side and have you think about that. You do not need to know all of these steps here on the left for glycolysis. I just wanted you to understand that as the structure changes, we basically create energy as we go through each chemical reaction. And anybody have a guess as to what is running that chemical reaction? What would be behind each of those green arrows from a reactant to a product here in glycolysis? Think about what we went over on Monday, what you were studying at the beginning of chapter 4. What facilitates chemical reactions in cells?
The enzymes.
Enzymes, exactly. So each of these steps is catalyzed or facilitated by a very specific enzyme. So you can count 1, 2, 3, 4, 5, 6, 7 enzymes that are going to be involved in glycolysis. So it might help you think about the role of enzymes a little bit more clearly, where in each step we need a different enzyme that's going to be able to use its active site to bind to, first one that binds to glucose, and then the product is glucose 6-phosphate. So the active site can't bind to it anymore, the product gets spat out. And then another enzyme that can bind to glucose 6-phosphate, and it destabilizes the bonds, and it rearranges to the product, fructose 6-phosphate, and so on until we have pyruvate molecules. And then when we look at aerobic respiration, the 3 steps that we look at are pyruvate transformation. So changing the pyruvate molecule such that it then can cross the mitochondrial membrane and get pulled into the inner interior of the mitochondria, into the matrix. And then there are a whole bunch of enzymes through a multi-step process of the Krebs cycle or citric acid cycle. And you can see we go to having different products. Citrate 6, C6, ketoglutarate C5, succinate C4, fumarate C4, oxaloacetate C4. You know why it's called the citric acid cycle? It's because citrate is used to make citric acid, and so that's where the term comes from. You don't need to know all of these steps, but what you do need to understand is what goes in, where the process occurs, and what comes out. So we make NADH. We pull CO2 off as a waste product because as we break down the original pyruvate molecule, we pull a carbon off and we make CO2, and we pull another carbon off and we make CO2 again. And we continue making through subsequent steps energy carrying molecules. And so you can see for every pyruvate molecule, we make 1, 2, 3 NADHs, and 1 FADH2 and 1 ATP. But for every original glucose, how many times does this cycle run?
2.
2, right. So for every glucose, we make 2 times 3 or 6 NADHs. We make 2 times 1 or 2 FADH2s, and we make 2 times 1 or 2 ATPs. And so the net from a single glucose molecule to the point where we've gotten the pyruvates through the citric acid cycle is 6 NADHs, 2 ATPs, and 2 FADH2s. And so we send those, plus these 2 NADHs over here, we send all of those to drop off hydrogens and electrons to the electron transport chain. And the electrons go from higher energy to slower and lower and lower energy as they get bounced from protein to protein embedded in this phospholipid bilayer. And as they get bounced down and have less and less energy, the energy that they lose actually lets these special channel proteins pull hydrogen ions against their concentration gradient. So from low to high concentration. And so what happens inside the interior of the mitochondrial membrane in between these layers is that we get a huge buildup of hydrogen ions. And so we not only get a concentration difference that's big, lots of hydrogen in here, very little here, but what happens to the positive charges? We also get a huge buildup of positive charge in here and a small amount here. And so then, see this kind of a slightly different looking molecule over here on the right hand side. This one that I just managed to block out again. I'm not sure what I did in my settings for that. This is ATP synthase. It's a special protein that's embedded all along many thousands of times over inside a single mitochondrion. And what happens is the hydrogen ions then flow through the channel of the ATP synthase protein. And that clue, synthase, that it ends in ASE, lets you know that this protein is actually an enzyme, and it's going to catalyze the binding of ADP and phosphate to make ATP. And this protein actually spins in the membrane. Literally, there's video of it. It spins as the hydrogen ions move through, and the ADP gets phosphorylated into ATP. And so the end result of all of this, from all of these energy carrying molecules, is that we make ATP. And it doesn't stay in the mitochondria, it gets sent out of the mitochondria to different organelles and to locations in the cytoplasm, to the plasma membrane, in order to attach to proteins and other structures in order to destabilize them and allow them to change shape and participate in chemical reactions. And so in a nutshell, this whole diagram walks you through glycolysis and aerobic respiration. So I'm going to, just so I can have peace of mind, she said, yay, what do I have, a budding chemist down there? Okay, so I'm going to hide this one and open up this guy and just take a minute here to just pause on a couple of images, okay? So this particular one that we have, here is showing you ATP being converted to ADP and back again. So if you look here, here's our full ATP molecule. So it's adenosine, which is actually a nucleotide, and it has a sugar, and then it has 1, 2, 3 phosphates, okay? Which is why it's adenosine triphosphate. And so it's actually a nucleotide as far as macromolecules go, but it doesn't make a full polymer because it doesn't make a long chain. This is the molecule by itself, okay? And what happens is, you see this minus delta G right here? When a phosphate is released, there's energy released with it when that bond is broken. And so when that phosphate can be transferred to another molecule, it can take that energy with it. And so ADP has less energy than ATP. And just, okay, I got ahead of myself there. Must have hit it. Okay, so here we are looking at ATP to ADP. Everybody can hear me, right?
Yes.
Okay, good.
Yeah.
And basically what happens is when ATP goes to ADP and back again, that basically when that third phosphate comes off, there's a whole bunch of energy in the bond between the second and third phosphate. And that energy that's released can be used to do work. And the phosphate being attached to another molecule then can make it unstable and likely to participate in a chemical reaction. And then when we invest energy into the system, you know, when we're inside the mitochondria and those hydrogen ions flow down through the ATP synthase, the movement of that enzyme actually can bind the third phosphate back to the ADP and make it ATP again. And so it just goes through this continuous cycle of losing a phosphate group by transferring it to another molecule, having a lower energy, and then going back up to higher energy and around and around. And each time an ATP releases a phosphate and gives up some energy, it's much less of an energy shift than if a whole glucose molecule was to be burned up right at once. And so the cell's then able to use little bits of energy to perform chemical reactions, as opposed to, you know, you can drink out of a glass of water or out of a garden hose, but how would you like to try a fire hose? It wouldn't work so well, right? And so it doesn't work for the cell either. And this slide just shows you the same kind of thing. If we burn glucose, we would, and we burned it all at once and made water, we'd have a whole bunch of energy released that wouldn't be useful to the cell. But when we make ATP in the electron transport chain, we're able to make it a little bit at a time, and then we still get the water at the end that you recognize as a product, and we still need oxygen available in order to make that product, but it's smaller step by step, and it's very controlled by the cell, and that's what the mitochondria let a cell do. In prokaryotes, they don't, there's a limited number of them that perform anaerobic respiration, which is further breakdown of pyruvate in the absence of oxygen. Then there are prokaryotic cells where all they can do is glycolysis, and then they have to burn tons of sugar compared to a eukaryotic cell in order to make the same amount of ATP. Now, sometimes I wish I were an inefficient prokaryote, because then I could eat all the junk that I want without storing the excess energy on my body, but that's not the case. Okay, so this diagram that's in here just shows you where things are occurring. So here we are in the cytoplasm, and what process happens there? Glycolysis. And we go from the glucose as the substrate to making pyruvate or pyruvic acid. It's two words that mean the same thing, as our product. So that's glycolysis. And what is it that we net from here? 2 ATP and 2 NADH. Now, then what you don't see right here labeled, there's another process, and this is called pyruvate transformation. Okay, and so it's like aerobic respiration, step 1, and then this Krebs cycle or citric acid cycle would be step 2, and then right here, the electron transport chain and ATP synthase action, which we also call combined oxidative phosphorylation, would be step 3. And see how we show those 2 ATP made from the Krebs cycle, and we show those 6 NADH and 2 FADH2 here. And we also are showing the 2 NADH that get made when pyruvate is transformed. What you don't see at this diagram are all of the inputs and all of the waste product outputs, but it does show you very nicely glycolysis before, changing pyruvate into acetyl coenzyme A to enter the mitochondria, and then the things that happen inside, and basically what you net out of the whole thing. So that's why I have that slide in here. These slides here just basically explain, this one explains glycolysis that I've walked you through, so I would read through it again. And this basically explains how the ATP gets used initially to activate glucose and make it unstable. And then this diagram shows you in a nutshell what I went through before on the initial screen that I showed you, where 2 ATP are invested, 4 ATP are harvested, so you net 2, and that you get to an ADH. And what this slide also does really well is show you basically what goes in, so before the arrow, so these are all what's going in, and then at the end of the arrows, right, these are showing you what is coming out, okay? And then at the bottom, we have overall what goes in and what comes out, okay? This one is just similar to the slide that I have blank at the end, where it basically shows you in a eukaryotic cell, what are the 2 pathway choices if the cell can actually conduct fermentation? Well, if oxygen is present, then where will the pyruvate go? It'll get changed into acetyl coenzyme A. It'll get pulled into the mitochondria, and aerobic cellular respiration will occur. If no oxygen is available, and it's the type of cell that can do it, then in muscle cells, for example, you would have lactic acid fermentation, and you would have lactate as a product as evidence of that. Or when we look at cells like yeast cells that are fungi that can do this, when we deprive them of oxygen in huge vats, we can get them to make ethanol and CO2, and that's where your beer and your wine come from, is by forcing yeast to go through fermentation by depriving them of oxygen. But most of your human cells, it's glycolysis and then followed by cellular respiration, and that's it. It's only in a few specific cells where we have the ability to do lactic acid fermentation, which is why it really becomes a problem if you are deprived of oxygen for very long, because what comes to a screeching halt?
Life, as we know it.
Yeah, because then suddenly all of your cells can't do what?
They can't get the HNAD, right?
Well,
NADH.
Yeah, they can't. Basically what happens is when we get to this end product of ATP, we recycle the NADH to NAD+, and glycolysis can keep going. But if we don't have that available, then we end up with just a few ATPs for a while, and then ultimately that's not enough. And so if you're oxygen starved, what really happens is it's not that you need oxygen that is used specifically in a chemical reaction. The oxygen is dumped on at the end and makes water. It's that when the oxygen isn't there, we can't make ATP, so we can't do cellular work. So why is it that, we're so worried about oxygen? It's because we need oxygen in order to keep this whole process going. And why is it that you can be like brain dead, but then your muscles don't like stiffen up till later when somebody, experiences the end of their life? It's because the muscles can continue within the absence of oxygen doing lactic acid fermentation. It's the lactic acid buildup that contributes to rigor mortis. So just kind of a dark aside, little enlightenment there. You really don't want to run out of oxygen. You really want to breathe. Don't ever let kids think that it's fun to do any of those breath holding YouTube exercises. Those are dumb. Okay, so a lot of this is just walking you through what I've been through, and I'm going to pull up and it explains what is in the diagram. So I'm not going to read it to you, but I did want to pull this one up and show you. So here is the 3-carbon pyruvate that you get at the end of glycolysis. And the reason we call it acetyl coenzyme A is because we use a molecule referred to as coenzyme A, and it enters the reaction, and it changes the pyruvate and it binds to it, and in the process we release a carbon molecule attached to some oxygen as carbon dioxide. And we actually don't keep the coenzyme A, it gets recycled. Think about the enzyme, okay? It gets reused. And so we change this to acetyl coenzyme A. It gets pulled into the mitochondria, and immediately the acetyl coenzyme A, the coenzyme A gets kicked off, and we have this 2-carbon molecule that goes through the Krebs cycle. And the coenzyme A stays out here, or gets kicked back out of the mitochondria and attaches to the next pyruvate molecule, and so on and so on. And so here's step 1 from that other diagram, where through pyruvate transformation, we make acetyl coenzyme A, and that's the molecule that can come across the mitochondrial membrane and go through the Krebs cycle. And so here is 1 carbon, and then we make 2 carbon dioxide molecules as we break down each of these. So if you look at it, here's 1, right? And here's 2, and here's 3 to count for all 3 of these guys, okay? All right, so what comes after that? So that was basically, here's step 1, here's step 2, and then what is this overall from the other diagram? This is step 3. And so you have a blank one of these to practice on, right? That was in that form, the Word document that I sent out. And so if you practice it blank, now this is where you're going to breathe a sigh of relief. If you had me face to face, you'd have to have this memorized and fill out the blank diagram and then explain it. Just use the diagram as a tool and practice filling it out and writing down rather than just staring at it and reading over it. If you practice on the blank one, it'll help you understand the process more thoroughly just by the process of writing things down and going through the steps, okay?
Is that in the module?
That is in the module. Remember where I showed you on Monday where I put all those resource files together above the lab? That's where it is. Yeah, it's in there, okay? And so once we get through pyruvate transformation and we get through the citric acid cycle, we have no more carbons. Those are gone. What we have is the products from glycolysis and pyruvate transformation in the citric acid cycle in the form of NADH and FADH2. And what these temporary energy carrying molecules do is release their electrons, okay? And they release hydrogen, see, because there's hydrogen bonded. And the electron transport chain is a series of specific proteins that move the energy. They bind to the electrons and the electrons get cycled through. And as the electrons move, their energy and their movement gets lower and lower and lower. And so when the electrons get kicked out at the end, they have less, they're still electrons, but they have less energy than at the beginning. And they combine with hydrogen and with oxygen to make water as a product, okay? And as they get pulled through, what they're also doing is pulling through hydrogen. And so where in the mitochondria do we get a high concentration of hydrogen? In the
matrix.
In the intermembrane space. Here we have H+. My writing hasn't really improved. H+ is high here, and then in the matrix, all the way in the interior, the concentration of the H+ gets low, right? And so now we have lots of charge and lots of hydrogen ions themselves. And so between the intermembrane space and the matrix, there's a concentration gradient and a charge gradient. And so now these hydrogen ions, as you can see by the arrow that was already there, they flow down. This thing actually spins. And as it spins, it binds ADP and phosphate into ATP as a product, okay?
The ATP synthase enzyme, is that are the hydrogens going down through it? Or
yeah, they're flowing through. It's a channel protein.
Is that facilitated? Is that active or passive? Would it be passive because it's
that's gonna be passive transport because it's down a concentration gradient. That's great.
Okay, thank you.
And so it's basically using the gradient to do work to make this ATP, and then this ATP where it gets sent. I don't really want to write elsewhere. It's too long. It gets sent out, right? And when I say to do work, it's not that the ATP by itself can do the work. It's that it's used to couple endergonic chemical reactions and convert them and make them exergonic so that those reactions can actually occur, okay? So this part is the electron transport chain. This part is the ATP synthase. And together, do you remember what we call them both?
Oh gosh.
We call them oxidative phosphorylation. And the reason it's oxidative is because it happens in the presence of oxygen. And the reason it's phosphorylation is what gets phosphorylated, what has phosphate added to it? The ADP. And so somebody decided to make your brains ache and call it oxidative phosphorylation. Another term you might have read is chemiosmosis. Now remember, osmosis is the movement of water down its concentration gradient. Well, chemiosmosis is referring to hydrogen ions moving down their concentration gradient. That's all that is. Just another word that if you see it, don't freak out. Okay, so this slide explains how those NADHs and FADH2s drop off their electrons and their hydrogens to the electron transport chain. And then this slide explains how that creates a hydrogen ion concentration gradient and what happens, and how if there was no oxygen, it can't move. You know, the electrons would stop, the electron transport chain would stop, and then we wouldn't make ATP, and then whoops, the cells would be in trouble. Okay, so it's in writing, and this is where you see chemiosmosis, and it explains how there's the electrical and chemical gradient of hydrogen ions and how they flow back down. So I think when you reread those slides now, you'll find that they make a whole lot more sense. And then this slide just summarizes molecules that go in and molecules that come out, starting with glycolysis and working through aerobic respiration, okay? And this, it took 2 slides to put that in, to fit it all in, okay? So that's what that one is. And then here's just a different way of looking at it altogether that's a little bit different from the slide that I showed you already, but it basically walks you through and it shows you, you know, where we get ATP from and how that all adds up to approximately 36 to 38. And the reason there's some variation is because it really depends on how much energy those electrons have. And the more energy they have when they get dropped off by the NADH and FADH2, then the more hydrogen ions they can pull through, and then you can just make just one more ATP molecule if the electrons have a little bit more energy. And so that's why there's some variation there, okay? All right, so fermentation, as I mentioned already, fermentation, the only reason it exists is to regenerate NAD+, okay? And that's what this is right here, okay? And what we already went through right here, that if you run out of NAD+, what happens to glycolysis? It stops, and that would be really bad news because the organism would die. And if you're the organism, you don't want that. Okay, this one just walks you through what lactic acid and fermentation and alcohol fermentation are as far as producing products.
I have a question.
Uh-huh.
So you were just talking about, so there's a question on the quiz. Why does death result from any situation that prevents a person from breathing? And I keep answering glycolysis requires oxygen.
Nope.
The other 2 choices are lactic acid builds up in cells causing death. I can't imagine that that's what would cause the death. And then the electron transport chain will stop moving hydrogen ions and no ATP will be made. Okay, now I'm thinking it's that one.
It's that one because it's this right here. We need oxygen right here. And if there's no oxygen, this gets cut off.
Okay, got it. Thank you.
And if this gets cut off, then we can't make ATP. Yeah.
Is that the pretest that you're referring to?
Yeah.
Okay.
One of the practice quizzes? Yeah.
Yes. I'm not doing well on it yet.
Okay, well, there's a couple there's 2 of them in there, right? For this week and then 1 from last week, I believe. Or is it 2 from last week and 1 from this week?
Yeah.
I don't even.
Yes, it is.
Okay.
Are they in the same
so-
place as the other resources in
Yeah,
the?
yeah,
okay.
they should be at the top of each of those modules. There's 2 at the top of 1 for last week and 1 at the top for week 5. Okay,
Yep.
and they're just practice quizzes. You can take as many times as you like. Okay, so then basically when we look at fermentation and why it's necessary, we see here's the process of glycolysis. and we make the pyruvate. But if no oxygen is present, then what happens to the pyruvate? It doesn't get used. But then if we've made NADH and we use up all of the NAD+, if we use all of this up, then glycolysis stops. And so even though the pyruvate is going to get wasted, we have to burn it up and make a waste product, which is what lactate or lactic acid and ethanol are. They're waste products to those cells. But what happens is we make this NAD+, and you see this big arrow here and it says regeneration. We enable glycolysis to continue because we provide the NAD+ that's necessary in that set of 7 chemical reactions that I showed you at the beginning. So this one shows you that we get a 3-carbon molecule that's lactate or lactic acid that's a waste product. And when in cells that can do alcohol fermentation, you get ethanol and CO2 as waste products, okay? That was the end pretty much of the PowerPoint, but I wanted to go through this one here because it's going to be on the exam as a multiple choice question, and I want to see if you can figure out the processes here, okay? So if you look, the process of converting glucose to pyruvate acid or pyruvate, what would we put in here? This is
Glycolysis.
Glycolysis, yes. Oh, this is going to be really bad. Okay. And if no oxygen is present in cells that can do this, we're going to have fermentation, right? So B and C are both what? They're both the process of fermentation, where B is specifically lactic acid fermentation and C is specifically alcohol fermentation. So remember, this is only, fermentation is only in cells with that ability. So I'm going to put only in some cells, okay? So in humans, it's muscle cells. So your brain's in trouble, but it's part of the reason evolutionarily why that exists is we can keep the muscles going for a while and we can divert oxygen to the brain, but then eventually your body says, stop, you dummy, you need enough air, and so you have to stop exercising so you have enough oxygen so that your mitochondria can all have the oxygen that they need. Okay, so if oxygen is present, then in a eukaryotic cell, what is the first thing that happens to the pyruvic acid or pyruvate? Step F is pyruvate. That's an A, believe it or not. Pyruvate transformation. Maybe I should have added a text box, huh? You know who would get a kick of my scribbly writing? Get a kick out of it? My mom. She has Parkinson's. And so writing is sort of a thing of the past for her, and it's with her left hand, and it looks pretty frightening. But probably about as good as this.
So for some reason, earlier in this hour, I wrote energy investment phase above this chart. Is that correct?
No.
Okay.
This is just the big picture of metabolism. Okay. Glycolysis, fermentation, and aerobic respiration, okay? So over here, fermentation, up here, glycolysis, over here is aerobic respiration, okay? So F is pyruvate transformation. D, I'm going to abbreviate as the citric acid cycle or Krebs cycle, okay? And then E, once we're, and so we've pull the pyruvate in by converting it into acetyl coenzyme A through pyruvate transformation. Now the acetyl coenzyme A enters the citric acid cycle, and when it's done, we make more NADH and FADH2, and those enter. Do you remember what this one is called?
Oxidation something, phosphor.
Oxidative phosphorylation, and that consists of the electron transport chain and
ATP synthase.
ATP synthase. Okay, feel pretty good about that now? So the ovals and the letters, those are just the names of the processes to tie metabolism together from the point of glucose. Take a nutrition class, like a biological nutrition class, and you could learn about like everything that happens before and after. Okay, so this is a diagram that is blank in the handout. And how would we fill this in if we were going to label it? So just, this is why something like this is helpful. Where is this orange rectangular box? Where's the location?
Glycolysis?
Now, where are we in this big box?
We are, well, there's the mitochondria there.
So if we're talking about, you know, the cell, this
Cytoplasm?
is, yeah, this is the cytoplasm. And what is this kidney-shaped thing?
Mitochondria.
The mitochondria, right? Okay. What are these bright yellow star-shaped things?
ATP.
Yeah, how much ATP we're making. And each of these is a process. So this one is going to be glycolysis, right? So I'm going to put the G. And this one is step 1 of aerobic respiration, which is pyruvate transformation. That's an awful P. Okay. This one is the citric acid cycle or Krebs cycle.
CAC or?
Yeah. And this one is oxidative phosphorylation, which is the electron transport chain and the ATP synthase action. So a challenge for you would be, can you review and remember what the reactants are and the products? And then that product becomes a reactant for the next one. And then what is the product here? And could you remember what the main products besides ATP are coming out of the citric acid cycle, particularly what's going on here? There should be another orange box right here that's joining up with this. So here we make
NADH.
NADH. Here we make
NADH plus.
NADH. And here we make more NADH.
And FADH.
And we make FADH2. Because there's room for 2 hydrogens to bind at the end of this molecule, right? And so all of those dump their electrons and their hydrogens off at the electron transport chain, okay? And remember, going into this, we also have oxygen, and we get out water, okay? Remember that down over here with pyruvate transformation, you make CO2, and over here at the citric acid cycle, you also make CO2. Going into all of this, there's some water that's used. And so this is just a great place for you to review. My red writing all over is really terrible and messy. I would challenge you to take one of these and see if you can walk yourself through the process put it, put this diagram, draw it in your study guide, because then you'll have it in a place where you want it, where on the test, because it's not closed note, it's just how fast can you refer to, what's outside your brain in addition to what's in your brain, right?
So the oxygen is coming in, and then the electrons and the H plus is going out.
No, so there's two parts, remember? Here. So here we have the NADH and the FADH2, and they drop off hydrogens, right? And they drop off electrons, and those get pulled through. And so then here we have electrons and we have oxygen, and for every half of an oxygen molecule, we also use hydrogens, and so you need 2 hydrogen ions, 1 oxygen atom, and an electron
To make water.
to make the water. And so if there's no oxygen available, remember, this will stop. Okay? And then this is where these are hydrogens again. So see how this just gets you to practice? Hey, do you know where this is? Do you know where this is? Do you know where this is? Do you know what this thing is? Do you know what this thing is? So practice them. And if you practice it once or twice and talk yourself through the process, then when you take the test, you will know exactly what goes in, what comes out, where it's located, and how it works, just by practicing the diagrams. The diagrams will pull and deep layer those processes into your learning, as opposed to just staring at the screen and reading and reading and reading, or looking at it labeled. That's why those blank diagrams are really, really helpful. All right, so that I gotta escape. We'll keep my annotations for now, and I can save it as something else. I'm gonna close that. So in the blank diagrams, you have just, you have a slightly nicer one here at the end. Do you see on the right side of my screen? That has like the water molecules and the carbon dioxide. And so this would be another one that could really help you thoroughly label, okay? And you might actually see this one, hint, hint, pulled into your test, where I ask you to tell me what's happening in stage 1 and, stage 2. And I've got to get a little text box in here that shows you, because this is the pyruvate transformation. So understand what's happening in those. Oh, the one that we didn't go over in the PowerPoint was this one that was about energy coupling. But I believe that one of the Bozeman videos goes through that really, really well. Is that sounding right to anybody else?
One of them does, yeah.
Yeah, I
Yeah.
remember seeing that.
One of them does. And it's not in this one. I was talking about it briefly on Monday. It's in the other video, not video. It's in the enzymes PowerPoint. It's slide number 11 right here. All right. And so go back and look at the Bozeman science one on enzymes and reactions, and he walks right through this. Okay. The only other thing to talk about today would be the lab. Does anybody have a question about the lab?
Do I have to do it?
You get to drop 1 lab score, but not 1 lab test score. So no, you don't have to.
Well.
No, you don't have to do it. So, but
No, I haven't looked at it yet. I printed it.
Make me log in again. I'm not sure if it's going to show me. Okay, good. It's not going to show you my password. Okay.
Will you have a Zoom meeting Monday night, even though it is a holiday?
Well, it's scheduled. And I don't know, yeah, it's a holiday, but this is remote learning, so it's
Yeah.
kind of like it's not a holiday.
Okay, well,
And
I guess-
so if people, I will be there, and I'll make a recording.
Okay.
Here,
it might be useful.
okay, I turn the recording back on, and I am going to put it on one page and make it bigger. So basically, the goal of this week is to use this online simulation and basically to learn about enzymes. So a lot of this background information, that's just, if you've watched a couple of videos by now, then you should be able to fill this in. And this would be really, really important information to have for you to do the test, because it reviews for you what enzymes are and how they function. Okay, so that's just that first page. And in this particular experiment, you're looking at how lactase as an enzyme splits lactose, a disaccharide, into D-galactose and D-glucose, specific isotopes of galactose and glucose. And basically, you're just simulating adding lactose powder to test tubes with lactase enzyme and water, and you're just measuring how much glucose and galactose are produced in different environmental conditions. And so if I open this Flash player for you, I'm going to laugh if it decides not to run on my computer right now. Because it was running earlier. All right. Adobe Flash is going to get retired. So there are, there are virtual labs that eventually nobody will be able to do. And you can see that it's pretty basic here. And you got a little journal. And if I split my screen here, and I'll put the simulation over here, and the directions here. You have to write a hypothesis. You have to come up with the independent and dependent variables, and then you have to fill things in. And it wouldn't be that hard for me to make a Zoom recording where I actually just walk through all of it and I could post it, but then everybody'd have to fill in their table and so on from me running it. And all you're really doing is, dropping the stuff in here and watching it, run a timer and then figuring out what happens, okay? So that's all that's happening in the lab. And then you're writing down, what you think is going to happen with each of those. And if you look at the lab, they're all in a pH of 7 right now. And we have a small amount of glucose, half a gram, 1 gram, 2 grams, 4 grams, and 8 grams. So it's basically doubling. So is there going to get to be a point where at a neutral pH, the enzyme can only break down low lactose at a maximum rate, and it doesn't matter how much more there is? So if all the clerks at Walmart were busy at their checkout stands, there would be a max rate that they could process, customers and get their groceries and whatever else they were buying scanned and bagged. Whether there was, 30 customers or 300, there's going to be a max processing rate. So you would explore that here. And then what do you think would happen if we changed the pH? If an enzyme works best at a specific pH and a specific temperature, then those are going to be impacted by being changed. And so you graph the regular data at a neutral pH, and then it says you clear your simulation, and then you're going to look, that one is substrate concentration, and this one is pH. So the first one is pH, and then the second one is actually what I was talking about with substrate concentration. And then you run it a second time with a different independent variable. And then you need to answer some questions about pH and temperature and, you know, substrate concentration and how those affect how the enzyme works. So
the one you're showing right now is for when we're measuring the effect of the substrate, not the pH.
yeah, you know what
Okay.
I think I'm going to do is I will go ahead and tonight I will do a solo Zoom, and I'll have the simulation and the lab pulled up side by side, and I'll just make a half-hour Zoom to walk you through it.
If I've already done it, can you change things?
If you've already done it, yeah. If you haven't submitted it, or even if you have, just submit a
Resubmit?
different, just resubmit and put a comment that says, you know, grade the second one or something like that.
Okay. Thank
Okay, you're welcome. All right, so I'm still recording. I had to remind myself whether that was being recorded. It's going to be
So
a
is the-
long video.
Is the test going to be a similar number of questions and minutes as exam one? Or?
Yes, yes,
Okay.
yes, it is. Okay, so the last thing for today was, this is the second, there are 2 learning assignments for respiration, and this one is just basically walking, this is the second one, and model 1 is walking you through looking at the steps. The link reaction is referring to pyruvate transformation. And so we've got glycolysis, pyruvate transformation, the Krebs cycle, and oxidative phosphorylation. And it's just having you walk through it as another good practice, okay? And then looking at what happens to the electrons and, you know, where are they coming from? And it says here, the reactions in model 2 are missing very important particles, electrons. So model 2, this is model 2. You're just supposed to fill it in, right? And so you might have to go back to the slide, the PowerPoint, and look up, you know, how many electrons do you need, right? Then model 3 is looking specifically at the electron transport chain. And so these questions are walking you through what's happening in the electron transport chain. And then model 4 is looking at ATP synthase action or chemiosmosis and walking you straight back, you know, straight through that. And then model 5 is having you review fermentation. So can you see how all of these are going to help get you ready for the test when you come? You know, it's basically walking you through studying. And if you're stuck on a particular step and it's something that you can't remember, go back and pull the PowerPoint up. Fast forward through this video. I'm going to convert it right now and get it up and, for you so that it'll be there.